Good morning, everyone. It's really an honor that I was invited to speak today here at this educational session about single-cell sequencing, and it's even a greater honor that some of you made it this early, 8:30. This is incredible. Thank you for showing up. And in the next 45 minutes, I want to tell you a little bit about how I, as a clinical geneticist, ended up doing single-cell RNA sequencing. And that's why I gave my title a slightly different talk called Next Generation Phenotyping. And I'm going to talk a bit about different projects, but the main thing is our recent single-cell perturbation screen. These are my conflict of interests, and I would like to start by showing you this slide, which is really one of my favorites, because it shows human embryonic development between, see how this works. Between day 22 and day 44, basically, we call this the organogenesis. And from this primitive streak here, all the internal and external organs form. And then basically the embryo is complete, and it just needs to mature over the next few months. And we learned yesterday that some people can study this actually in vitro from the incredible work from Magdalena Zernicka-Götz. But me being German, and also with the regulations here, and also me personally, I cannot and don't want to work on human development, even though as a human geneticist, this is where all the magic happens. So if we study malformation syndromes, now this is really what you want to study. That's why in my lab, we, for many years now, used the mouse as a model organism to investigate embryonic malformation syndromes. And what I'm showing you here in this beautiful picture is mouse organogenesis, and as you can see, it's much faster, just between day 9 and day 13, so mice are pregnant roughly 20 days. You see that this tiny thing, which is pretty much just a head and a heart, develops into a completely matured embryo. And what I'm staining here is, or what I'm showing you here, this beautiful blue staining, is one of my favorite genes called HOXD13. And the problem with this is this technology called in situ hybridization is, I would say, from the 70s and looks beautiful, but it stains one single gene. And as you know, there's many more genes. So looking at this organogenesis the conventional way is quite laborious. But I want to take the first few minutes and tell you a little bit about my background and why and how we can use the mouse as a model organism, and then why this is not sufficient all the time. Good. As a clinical geneticist, I think almost 10 years ago now, I saw a family, or actually we had several families, that had something that we call a brachydactyly, and you see this here. The thumb is short, and also the index finger. And at the time, we found a deletion of a gene called FRNA4. So I was very proud. I thought we found a new disease gene, and we knocked it out in the mouse, and absolutely nothing happened. But then we learned about something that now is more or less standard knowledge called topologically associating domains. And what you see here is embryonically or developmental genes are surrounded by enhancer elements, non-coding elements, and they are in these regulatory domains, and they keep looping towards their target gene. And for example, this gene here is expressed in the limb, and a neighboring gene is expressed in the brain. It has its own enhancers. And then they're separated by so-called boundary elements. And when I first learned about this over 10 years ago, I thought maybe this could relate to my patients that I showed you before, these guys here, because the gene here wasn't the disease-causing one. So when now with the knowledge of TADs, I looked at this very same patient, or these patients, and saw that, well, this deletion doesn't only remove the gene, but also the TAD boundary. And so we had this incredible hypothesis and said, maybe the deletion of the boundary causes the phenotype, misregulating the neighboring gene. And this is now old stuff and all published, but at the time, no one really wanted to believe us. So then we said, let's make a mouse model. But when we just knocked out FRNA4, like I said, nothing really happened. So then a lucky thing came along, and CRISPR-Cas was invented, obviously not by us, but we used it to generate deletions, inversions, and duplications at very high speed. So high speed means like 10 to 12 weeks. And this was a mile, like really much, much faster than previously, where it would take you 2 years. So what I'm telling you now is that with CRISPR, you can manipulate the mouse genome and many other genomes almost at will. And this makes it possible, and in this to recapitulate what human deletions or mutations are like. So what you see here is this was our hypothesis. We said by the deletion of a boundary element now, a foreign enhancer could come close to a target gene and starting hijacking it, causing misexpression in the limb. This gene should only be expressed in the brain, but now it's also expressed in the limb because it adopts or is hijacked by this foreign enhancer. And we wanted to test this in the mouse, and you see here, this is the normal limb of the mouse, and the neighboring gene, PAX3, is not expressed there. And if this is true, you have a beautiful hand in the mouse and in the human. Now we wanted to see if our mouse has the same phenotype here. So like the patient, so we deleted the boundary, and suddenly you see the neighboring gene being hijacked, and the mouse has exactly the same phenotype like our patient. So here's short thumb and short index. So we were very happy, and I should stop my talk here and tell you that in the following years we published many more cases like this, and we're very proud that we got on big covers of magazines. The problem is that not many people care so much about limbs like I do. I had to learn this the hard way. And when we now, the limb is great because mouse and human are pretty similar. But in the brain, for example, or other organs, this is much more difficult. So it's much more challenging to mimic intellectual disability or brain malformation in the mouse. And I've personally made a lot of mice that are very healthy, and that's good for the mouse, but not so good for the scientist, because you don't really understand. So that's why this is now basically just the introduction. I think the main challenge, or one of the key challenges for us now, is that we can do, as you all know, next generation genotyping. So we can sequence anyone here in the room very quickly. And now with CRISPR-Cas coming along, we can also do next generation genome editing. So we can rewrite the genome. But what is still very low throughput, by making one mouse after another, what we really need is next generation phenotyping. So we need to be able to read out a phenotype and not just look at the mouse from the outside or any cell line. And coming back to the initial story, so when a few years ago, I explained this problem to a guy called Jay Shanduri at the University of Washington and told him, well, this is a beautiful picture, but it just investigates one single gene. So wouldn't there be a more modern and faster way to analyze these embryos by just looking at one gene at a time, because this will take forever? And Jay said, well, maybe you come over to my lab and we try something new called single-cell RNA sequencing. And the idea would be we take these embryos into single cells or single nuclei and then analyze them, and then we can reconstruct them on the computer and do something that I like to call in silico developmental biology. And it seemed crazy at the time, but that's exactly what we did. So what about single-cell sequencing? And I'm mainly going to talk about RNA. So the main idea is, like I said, you have some organ or blood or even an entire embryo, and you dissect it into single cells. There's different protocols for it. You dissociate it. And then there's two major approaches. One is called the well-based technologies. So there you actually put a single cell into each well. And a more commonly used technology is a droplet-based. So there you still dissociate each single cell. And then you put them into a tiny compartment, in this case, an oil bubble. And then you put some chemicals on top. And then you can do genome sequencing, transcriptome, proteome, whatever. Like probably 5 papers are being published about this right now, about new approaches. The main drawback is that it's extremely expensive and the throughput is very low. So you get like 20 to 60,000 cells, and it's extremely expensive. So this was never going to work with an entire embryo. So that's why I joined Jay's lab and paired up with a guy called Jun Kao, and we developed a new technology. And I'll be quick on this, on the details, but the main idea here is that we never ever physically separate the cell, like here, because this is a major bottleneck, but we take a very conventional 96 well plate and take a few thousand cells, or in our case an entire embryo, in the first well, And in the second well, another embryo, and in the third, a third embryo. And then we don't take the entire embryo, but the nuclei, so the cells, and then we add a molecular barcode to them that is unique to the individual well. And then you mix and pool them, and you can do this on your normal bench, and add a second molecular barcode, mix and pool again, and use a third molecular barcode. And by this trick, you can actually do this even further, 4 times, that in the end, you end up with cells that are uniquely labeled, and by the combination of the barcodes, you can trace back which embryo they came from. And this was one of the first approaches how one could use single-cell RNA to actually profile over 1 million cells in a single experiment. And we did this in order to investigate my favorite time of mouse development, so organogenesis. And this is what the data looked like. So we took these embryos, dissected them in single cells, and then you get something that you call a T-SNE plot, a UMAP plot, for the people not so familiar with single-cell sequencing. What it means here is every single dot is the transcriptome of a cell. And for example, these guys here are all the epidermal cells, epithelial cells, sorry, of the entire embryo. And this are the hepatocytes, so they cluster based on their cell type. And when I first saw this, I went back to my colleague June and said, like, where the hell did my mouse go, right? It really doesn't look like a mouse anymore, but like modern art. But once you get into it, we could identify that there's over 500 different cell types in the developing embryo. This was nice, but, and this is what I always tell you, now we came up with the very cool technology to take something into tiny pieces. And let's compare it. Usually people talk about a smoothie. I like this one better here. So we have a car, which is nowadays very complex. And we now, by single-cell RNA sequencing, or we call it SCI-seq, single-cell by combinatorial indexing, we can now take this car apart, right, into all its original pieces. And this is really nice. So my daughter once did it to my phone. The problem is I could never fix it back together, right? It's easy with some things, so like this is probably the tire, but I'm not sure about all these pieces. So basically what I'm saying is we have a technology to see all the different pieces, but what do they mean in biology? That's more difficult. And for this, we need bioinformatics, and I cannot emphasize this enough. We all know that large data sets need great computational scientists, and we at the time paired up with a guy called Cole Trepnell, a single-cell bioinformatician, and he has an algorithm, or we developed it, where we can now follow cells through developmental time. And the idea is you have a cell, and at some stage it will make a decision to become that cell type, or in this case, go into disease or stay healthy. And wouldn't that be awesome if you could do that on your single-cell data set? And this is actually exactly what we did, and now I'm showing you a movie that has been shown at this conference several times, but I still show it. This is our initial embryo, UMAP, and there, for example, the hepatocytes are very close to parts of the brain, and I don't really know the origin of the different cell types. And that's why now what the algorithm does is it clusters each cell based on its origin. And now, for example, all these cells here represent the mesenchyme, and you can see through developmental time, these are the muscles growing out of the mesenchyme. And the large fragment here flying by is most parts of the brain. And this tiny part here is my favorite organ, the limb. Looks really boring, right? So this is not only a cool movie, but it really helps to identify new cell types. For example, this is the developing blood. And for example, down here, this intermediate cell type, we couldn't identify before, but now we know that it's part of the brain. That made it much easier. And why is this cool? Because now I can really do in silico biology and in silico developmental biology. For example, I can tell the computer, without killing any more mice, show me all the developing muscles. And the computer, in almost real time, shows me that this is part of the side of the embryo, and this is the pharyngeal arch, and then there's 2 ways to actually make mature muscle. And when I first saw this, I thought I'm going to win the Nobel Prize because I found that there's 2 ways to make muscle, but a simple Google search found out that this had been found out many years ago. One is through MIF5 and the other one through MyoD. I just don't know very much about muscle. But the cool thing is it took them 10 years to find this out, and we could do it with a few simple clicks. So that's basically the promise. And this is also the introduction of the stuff that we now need to do more advanced things. So we generated a wild-type atlas, this a few years ago, but I think it's still relevant because basically you don't have to do an in situ hybridization anymore. Where's your favorite gene expressed? You can just go online and search for your favorite gene, and it will show you basically which cell type this is expressed. But then following up on this work, a grad student joined Jay Shanduri's lab, and I was very much involved in this. So the idea was, could we now build a truly digital embryo, so through developmental time, based on published data? Because people did this, not just us, we just did the later stages, 13.5, but how about we could do this from very early on and then build a database where people could browse this. And this is exactly, so he hired a very gifted student, CX, and this is exactly what we did. So he downloaded all the data that were available, and basically from day 3.5, when the embryo implants, then he followed the cell types and how they develop. And then the coolest thing is he actually built what we call a or a tube map or underground map, starting from implantation, and now you can follow all the cell types and how they develop, and more importantly, which transcription factors influence the cell fate decision. And I think this is extremely powerful. And then the idea came up, could we now also do this in mutants? Because wouldn't it be great eventually if this is all wild type, if we could predict based on maps like this, what would happen if, for example, this transcription factor here was mutated? What would happen to the germ cells? Or here, which way would they go? So we decided to take this to the next level, because I wasn't so interested in just wild type, but as a human geneticist, I'm interested in mutations. So could we take this technology and investigate mutants, mouse mutants at scale, and not just one at a time, but many, many. So now I come to the more recent work, and the simple idea is, I told you earlier on, with CRISPR-Cas, you can basically generate a mouse mutant very quickly, and then the major bottleneck is, then you have to hire a PhD student who works on it for 4 years, who then looks at the brain, looks at the liver, looks at the lung, with all these old technologies. People can do it, and we still need to do it, but it's very low throughput. And now the new developmental single-cell atlases coming out, so what I just showed you, where we also contributed to, how about we could combine the two? We basically wanted to build a digital mouse clinic. Back in the days, or you can still do that, for example, in Munich, we actually collaborate with them. You can take your knockout mouse and they let it run through a maze, they measure it and all that. But we wanted to do this using our approach. And Jana, a very gifted PhD student, set out on this almost crazy quest, but she still agreed to it, and said, well, I don't have the time, or I'm only a single student, actually it were 3 students involved, but to do all these behavioral tests, but I will just use a single-cell sequencing. And the idea was we would want to see how scalable it is by using over 100 embryos of 25 different mutants. So this is already on bioRxiv. You can check it out if you want to. And this is our experimental setup. So we decided to take 4 different wild-type strains, because in mouse genetics there's differences between the wild types, and some actually show mutations, and some just ignore them and don't show, some show phenotypes if you make a mutation, and some don't. So the wild type are important. And then we chose 22 different mutants and overall 100 embryos. And the idea was to sequence not just one thing, but the entire embryo. And she went and did the experiment using the combinatorial indexing, and we generated one of those UMAP plots where you get the major cell types. So this was all nice because we had the wild type already, right? We had generated previously. And then the big problem started, because now we had a massive amount of data, and I thought you would just go through it, but the bioinformatics behind it was challenging. So I want to explain to you what approaches we've taken and how we also tried to validate them so that you could be using them as well in your lab. So the first and most obvious approach would be, let's go for cell type composition changes. In a mutant, is there a certain loss of a specific brain cell type or in the liver? And this you can compute. This is rather straightforward using single-cell data. And then in the second approach, more advanced, we try to go for the subtle defects that you were not able to see using conventional phenotyping by developing a score. And last but not least, we wanted to validate this in very challenging mutants. And we chose mutants of different severity. So we started with very, very severe. And going down to new mutants where we didn't know anything about the phenotype so far. And now I'm going to show you, sorry, I'm not clicking this machine very well. I want to show you first a view of the entire data set. And because this is very complex, let me be slow. So here you see all the different mutants that we've chosen. And then we wanted to see, are there cell types gained or are there cell types lost in them? Once again, without killing any more mice. This is all done in silico, and that's why we make pie charts here, right? This would be a gain, and this would be a loss. And this is the entire data set on top of each other. And then you see, for example, down here, there seem to be some losses, and up here, there seem to be some gains in specific cell types. And what you can also see, that seemingly unrelated mutants have shared phenotypes. So this is quite powerful. But because this is just a big heat map, and you probably can't see anything, let's zoom in on a specific mutant called TTC21B. And we saw here that there was a massive loss in the retina. And I said, we sequenced the entire mouse, so would we really be able to see something as specific as the retina, which is just a few hundred cells? So we looked inside. We actually did conventional phenotyping. And just by looking at the mouse, we could already see this is a wild-type mouse, and this is the mutant, and there seemed to be something wrong. But when we then did histology with some help, oh no, we actually zoomed in further, sorry. So this is the cool thing about the bioinformatic approaches. We don't have to kill them, but we can do all this in silico. So we checked out what other cell types are missing. And then here, this is the gain cell types in the mutant, and this is the lost. And if you take a look down here, this is retina, this is the lens, and also retinal epithelium. So it seems that there are several structures missing in this mouse. And when we then just did H&E staining, you see here, this is the wild-type eye, this is the heterozygous, and then the mutant, it's severely malformed. And there's still some cells around that are difficult to see here. So the technology works, and basically we can trust our data. So this was good, because in the beginning, people always tell you, this is just bioinformatics, right? Whatever does it mean? But we can actually validate our findings using conventional phenotyping. Now, the cell type composition changes, I think, is pretty straightforward. Let's move to a next thing, the mutant score. What you see here is work by Fanny, a very gifted PhD student from the University of Washington. And we sat down and said, well, we need a more granular way to look at the mutants, so to find very discrete phenotypes. And she came up with a score that she called Local Cellular Heuristic Neighborhood Enrichment Specificity Score, and short Loch Ness, right? And we're in Scotland, so that's important, but mainly also because you always need a fancy name if you want to publish something, right? But sounds good. And the Loch Ness score, how does it work? So the idea is that in a wild type situation, because we have the wild type and the mutant and we compare them, you would always have wild type cells surrounded by mutant cells coming from the mutant if there's no change. But then there's going to be specific regions where there's more wild type cells, that would mean there's a mutant loss, or then there's regions with more mutant cells that we call mutant gain. And we wanted to visualize this. And this is exactly what the Loch Ness score does. So this would be the normal wild type, and here are discrete losses, and here are discrete gains. How does it work, or how could you use Loch Ness? So first of all, you can throw in your entire mutant and see, is there much happening? And then you get a curve like this, and we see here we actually knocked out a few TAD boundaries, and they look surprisingly normal. They all have a bit of loss, but they all have similar mutant score. But then when we go down here, for example, this guy, a SOX9 mutant, has a very broad, so there seems to be some gains and some losses. This seems to be, again, pretty unremarkable. And also here, this one seems to be changed. And then you can zoom in, once again, without killing any more of them. And ask, so for example, in the TC21b, I showed you already that there is a loss in the retina. And indeed, our mutant score picks it up very easily. But then we looked at other ones. For example, this is a GLI2 knockout. And there was specific changes here, a loss and a gain, in the roof plate and in the floor plate. And this, we wanted to check it out. So then you can zoom in further. And what's even the roof plate and the floor plate, so this is basically part of the neural tube and is extremely important for neural patterning. So now you can tell the computer, zoom in and tell me, show me all the roof plate cells and the floor plate cells. And this is what you're seeing here. And then you can run the mutant score and it tells you that there's actually a bit of loss down here and a bit of loss down here and a big gain in the roof plate. So basically diving in further. And the reviewers obviously made us validate this, and we did, so we're very happy that this is a real finding. But now coming to the third approach, now we really wanted to challenge the system and say, well, we can find cell type compositions and we have a new score. Let's go to a very complex mutant that we've known, and this is work led by Jana and Varun, a computational postdoc in the lab. And so a few years ago, when I was still in Stefan Mundl's lab, we published a mutant that knocks out SOX9, but it's actually a regulatory knockout because the full knockout is lethal. And at the time, because the lab cares about limbs, we just published that there's a limb phenotype. But we knew there was more because these mice actually die. But as you know, right, if you publish a nice paper, then you don't really care so much about it. So we said, let's try and give it a shot if we can investigate this, because SOX9 is a pleiotropic gene. It's expressed in many tissues, I'm sorry. And what we've done in this mutant is that in the wild type, there's many enhancers of SOX9 that drive its expression in the gonad and the bone. And in this mutant, there's an inversion, taking the enhancers, doing enhancer hijacking, and putting it in front of an unrelated gene called KCNJ2. And this causes lots of trouble. So we took the embryo and sequenced the entire thing, and then tried to play with the tools that we just developed. So the first and most obvious thing was, if you look at it, there were changes here. These were the other mutants, and the SOX9, I already showed it to you, had a bit like a weird distribution. So it goes into the gains very wide, and also there's seems to be a loss. So now you can tell the computer, please zoom in, which cell types are really affected. And it turned out that all the bone cell types were decreased, so osteoblast, chondrocytes. You can see that here in this visualization. All the mature bone cell types were decreased, but an undifferentiated mesenchyme, precursor cell type, was massively enriched. So that sounded like maybe these cell types don't actually, they're developed, but they stall, so they don't really develop any further. So really a developmental delay, but how would you prove this? Good thing is, with the single cell data, you can play your magic. In this case, Varun did. And now we used something that is called RNA velocity, and let me walk you through it. So here we see all the cell types of that will be bone eventually and connective tissue. And in an RNA velocity plot, you can see which cell type will develop into another one. And here's the undifferentiated mesenchyme, and this will give rise to intermediate mesoderm, connective tissue, osteoblast, and chondrocytes. And now we were asking, what does it look like between wild type and mutant? And we could see that in SOX9 in red, most of the cells are actually located still in the undifferentiated stage, so replicating what we see down here, while in the wild type, they do develop nicely. So, and if we can visualize it even more or better if we do a density plot, and then you see in the wild type they basically develop nicely into their cell types where they should be, and in the mutant they seem to get stuck in this earlier stage. And this is something that's very difficult to find out using conventional phenotyping, because which marker are you going to investigate, or which slice are you going to look at under the microscope? So I think using this technology in a very complex mutant, we can show by investigating the entire embryo that at least the cells of the mesenchyme are actually developmentally delayed or might even get stuck in this early precursor cell type called undifferentiated mesenchyme. And because reviewers are always critical, you obviously have to basically validate what you see. And these are some RNA scope slides, and there you see in the wild type, SOX9 is expressed in the bone where it should be here, and the target gene of the enhancer hijacking is not really expressed there. And in our mutant, we see that, as predicted also by the RNA-seq data, KCNJ2 is now misexpressed, and SOX9 is decreased in its target tissue, causing trouble. And I don't have time, but Jana Henk actually showed this last year here at ESHG. We didn't just go into the mesenchyme and into the bones, but there's other phenotypes that you can explore that I don't have time to talk about today, for example, in the brain and other tissues. This work is online available, right? And I'm very happy to say that it was just accepted in principle, but it will come out soon. But I think with data like this, it's extremely important that you make them available. So also here we will launch the website. And I think the main idea was really to develop a framework that also you could use. So you don't have to repeat all of the hassle that we've been through, but potentially you could also investigate your mutant. And we are thinking about starting a collaboration actually with the German mouse clinic, where we want to do this at larger scale and making the mutants that they have generated available to everyone, basically online. Good, and then you can browse through your favorite mutant, always versus wild type, without actually any bioinformatical skills. Good. Now I have 5 minutes left, right? So I'll talk about one more story. I showed you now that you can investigate an entire mouse embryo, and this is really cool. But I recently moved to northern Germany, and a guy came to me and said, like, it's all nice, but I actually don't care about the entire mouse. I'm just interested in a specific region of the brain, the hypothalamus. And Jens Mittag is a thyroid hormone expert, and he wanted to investigate something that we call thyroid hormone loss. And this used to be very frequent in people basically that were born with low thyroid hormones, and you don't see that today anymore. But they don't just look abnormal, but also menstrual retardation is a severe problem. And we still don't really know why the absence of thyroid hormone causes this phenotype. And some years ago, Jens and his team had published that in the hypothalamus, there's a very specific neuron type called bulvominergic neurons, and they are slightly decreased. So he said, well, if you have that fancy technology now, can you take a look what's going on in the hypothalamus of my knockout mice? And Varun took up the challenge and said, sure, now we do a more targeted approach. And this might be very relevant to some people here because not everyone cares about the entire embryo, but maybe just the heart or just the hypothalamus. And so we did single-cell RNA sequencing of the hypothalamus, and then you see, like I said before, these are the major cell type. There's many neurons, oligodendrocytes, astrocytes, and you can identify them based on marker genes. So this is pretty straightforward. But now, Jens told us, I know this mutant. Just look for my favorite cell type and don't care about the rest, right? The phenotype has to be in the neurons because they have intellectual disability. So Varun did his magic and looked in the neurons, but there wasn't much different really. So these are actually the major cell types, sorry. And we saw that there was no big differences except here, which was actually an artifact. So you can see this is just from one replicate. But there wasn't really a massive decrease of neurons. And even when we zoomed in further by just subclustering the neurons, we identified many different neuron types, but we didn't see major differences. And so if you here compare wild type versus mutant, this is 28 cells, so it doesn't really count. So Jens said, well, your technology is not good enough, right? There's something wrong with it. The patient and the mouse has a problem. So we said, maybe it's worth, because we generated the data, to also look somewhere else. Don't just stick to your hypothesis. How about an hypothesis-free approach? And Jens doesn't like that word. For him, it's a swear word, hypothesis-free research. But we tried it anyway and looked where else could be a phenotype. So where are the most differentially expressed genes? And first he didn't believe us and did his conventional stainings and could actually confirm that there was no difference in most of the major cell types in the neurons. And then Varun asked, where are the major differences in terms of differentially expressed genes, because this is the cool thing with RNA sequencing. You can not only get the cell type information, but also what's happening, which genes are expressed. And then it turned out that the oligodendrocytes are actually mainly affected, which is this cell type. So we went back to Jens, the expert, and he said, I don't care about oligodendrocytes. Who does? And we said, well, let's check it out. And so when we subclustered the oligodendrocytes, we actually found a very interesting phenotype, seeing that there was a complete separation between wild type and mutant cells. So what does that mean? That means that there is oligodendrocytes, but they behave completely differently. So they express different genes. And so we were very surprised. And you can then ask the computer to show you some of those genes. So these are the majorly differentially expressed genes. And Jens and his team actually then validated this using conventional technologies. And what's the takeaway from this? So I think, and this is one of the strongest things about phenotyping using RNA sequencing, this will show you phenotypes that you never even thought about. And I think this can sometimes lead you into ways, like in this case, no one thought about oligodendrocytes before dealing with thyroid hormone loss. Good. So now coming back to the human, and I know I started by saying that I'm interested in human development, but talked half an hour now about mice. We've also done some work on human. This was actually still done in Seattle, because all the technologies I just showed you can also be used on human embryos. And Magdalena Zernica-Götz actually yesterday showed a slide in her recent Nature paper. They used skyRNA sequencing to profile their synthetic embryos. So this was very nice. But as a follow-up to our mouse atlas, June Kao, and I was only partially involved, I just wanted to mention it here because it's relevant to the society, I think, also generated an embryonic atlas of human aborted embryos from a human embryo database in Washington. And he took the same approach. And the cool thing is now you can browse all these data. So it's all publicly available, and you can check it out and see where is your favorite gene expressed if you're interested in eye genetics or if you're interested in heart genetics. So basically the data is all there. And we, in my lab, also now use the data to rank disease variants, but more will come of that later. Good. And then the standard problem is that people say that humans are not mice, and I agree. As you can see here, they look very different, but from the single cell perspective, if you overlay the data, they actually look incredibly similar. So here we see that the major cell types, neural trajectory, hematopoietic, mesenchyme, there's hardly any difference between mouse and human. And the major difference that drive the separation here in UMAP space are actually driven by the developmental stage, because the human embryos were later than the mouse embryos that we generated. Good. But also in my lab, we generated some human data using, in this case, not Sky, but we used conventional 10x sequencing, because if you work in very precious tissues, skyRNA sequencing is not the way to go, because you have a massive loss. So sometimes in an experiment you lose 90% of your tissue. And when I was recruited to Kiel and Lübeck, where I'm now in northern Germany, they have a strong interest in Parkinson. And I didn't know anything about Parkinson, absolutely nothing. But César, a great postdoc in the lab, said, well, we could just use it for unbiased phenotyping. And together with the Anne Grünewald lab from Luxembourg, we set out and got our hands on post-mortem tissue of humans that died of Parkinson and wild-type controls, so aged and sex-matched. And I know that there's much bigger consortiums going on, for example, in Leuven at the moment, but we had small numbers, and these brains were fresh frozen and then dissected. So we FACS sorted for the viable cells, and then did 10x RNA sequencing. And like I told you before, you don't need to know much about a tissue of interest, but you just do clustering. And this is what we did. And saw that there's all the major cell types of the midbrain, because these were tissue sections of the midbrain, including the substantia nigra, which is thought to be affected in Parkinson, because this is where the dopaminergic neurons are located. So we were happy to see that there's lots of oligodendrocytes, astrocytes, microglia, but there's also dopaminergic neurons, but very little, so just a few. So we were worried and said we did something wrong, but the slices were very thin. And it turned out that actually most of the dopaminergic neurons we didn't really get. But another interesting fact is that we were able to identify a cell type that had never been described before. It doesn't belong in the brain. And what's even better, we called it CutPS2 high because this is one of the genes that is expressed there. But when you now zoom in, we could see that this cell type, CutPS2 high, was almost exclusively found in the Parkinson patients and almost absent, not quite, in the wild type. And so these were individuals that did not die of Parkinson, but maybe there were some cells there. So now the question, and you can see this also quantified here, so CutPS2 cells are really, really specific to very few, I must admit, but really specific to the Parkinson brains. And so then, obviously, you have to validate these findings, and we could see that these CutPS2 cells actually come from the substantia nigra. And when we did stainings, we could also validate that these cells were actually the dopaminergic neurons that lost their ability to express dopamine. For example, these are dopaminergic neurons that are still able to express dopamine, and this is a CutPS2 neuron. So those are the dying neurons. So it's a cell type specific to Parkinson because these cells are dying. And because I'm running out of time, I will spare you with the rest and come to my conclusion. So I showed you that we now can do next generation genotyping. We all know that. Next generation genome editing. And I believe that single-cell sequencing is indeed the next-generation phenotyping strategy. And now, what we do in my lab and many other labs, we combine the two, or the three. So we generate large CRISPR libraries, transfect them into cells, and then do readout by single cell. By this, we can, from the thousands of variants we find in a patient, we make thousands of variants, and then we analyze them in one experiment, and ideally in vivo. I'll talk about this more next year because there's more stuff to come. And want to summarize, I hope I could convince you that combinatorial indexing-based profiling, you can do millions of cells in a single experiment. In our case, we did over 100 embryos. And it gives you sky-RNA profiling, or also ATAC, gives you unbiased phenotyping of not only entire organisms, but even the region that you thought you know really well, you might discover new things. And I personally think this is the future of pathology, right? If we do this on every single biopsy, this is going to be very, very efficient. With this, I would like to thank all the members of my lab. Today I talked a lot about Jana's work and also Varun's work, but there's many people involved in this, and our collaborators, Jay and June in the US, and also Cole Trapnell. Without them, it would have been impossible. Fanny and CX, who did some of the work. And our clinical team, and last but not least, I would like to tell you that in Schleswig-Holstein, which is a tiny state, there stands a new research building, beautiful, in Lübeck. And if you take a very close look, this is where we are located, there's an empty bench or an empty computer. So if you like what you saw, you can write me an email and maybe we could work together and you could join our team. With this, I'm happy to take some questions. Thank you.
Thank you, Malte. That was a beautiful overview. In the interest of time, we've probably just got, we could perhaps have one quick question if there's any out there. Is there someone going towards the microphone? Yep, I can see someone go to, no, you're leaving, okay. Okay, so Malte, I was just thinking about the comparison between mouse and human. You're quite right, there's a lot of similarity there, particularly in the coding regions. When it comes to non-coding, certainly my experience around SOX9, in a third of the cases, there's good conservation of those enhancers, but 2/3 of the time they seem to be very human-specific. So we're going to have to, at some point, have those human-specific models to untangle a lot of those non-coding aspects of the genome, which make the real difference between mouse and human.
I totally agree. I think much of the difference comes actually from the non-coding regions, but we also got very frustrated by knocking out non-coding elements in the mouse, and usually the mouse just doesn't show any phenotype, but this might also relate to the fact that you simply don't see it using conventional sequencing, conventional phenotyping. So we've now dug deeper into this, and you do see some transcriptional changes, so molecular phenotypes. That being said, I agree that we will not get around to it to actually investigate human enhancers in an in vivo context, and it's going to be challenging, but for example, synthesis we could humanize loci and investigate them, but there's clearly differences and much more work to be done.
I'd be keen to come up and work with you on that. All right, thank you very much. I think we'll have to close that session now.
Yeah, I'm sure Malte would stay around if you want to come up and ask some questions after the talk as well. So let's move on to the second talk in this session.